So what's the deal with GMOs? In this video, we're going to take a look at the DNA technology through the use of genomics and biotech that's allowed modern-day genetically modified organisms. So in this video, we're going to attempt to answer the question, what's the deal with GMOs? Let's get started. So before we can learn really about modern day biotechnology, we need to take a look at the history of genetic manipulation. So let's start with a general statement. Genetic manipulation of organisms is not new. We've been doing this for thousands of years as we've breeded different animals, as we've crossbred and hybridized plants. This is something we've been doing for a long time, right? So even researchers at the USDA can selectively breed carrots for color, right? Without any going in and tinkering with genes, we can just breed this carrot with this carrot and actually get these multiple colors of carrots just from selective breeding. We also, of course, have our modern day pets, man's best friend, you could say, our dogs, which have arrived to us as descendants of the wolf. So how do we get this large variation in this variety in dog breeds? Well, because we've selectively bred them to get the small little, dogs you put in your purse. Well, we've taken the runts of this litter, the runt of this litter, bred them together and take the runts of that litter. And you can see over time how breeding certain dogs together with different variations would give us some variety in our dogs. And then if you take a look at corn today, right, corn's been very popular lately. If you see the evolution, modern day corn actually was just from selective breeding from ancestral teosinte, right, this corn over here. So by selectively breeding for these kernels, for the size, we've actually got this sort of modern day corn. Now, so that's our history. We've been crossing and modifying genetics in our foods and animals for a long time. Well, what's different today? What's different is that we can modify at the molecular level. So we can actually genetically engineer today by changing or manipulating the DNA, which of course makes the animal or the vegetable, the food that we're looking at. So if we're going to start engineering DNA and genes and organisms, we need a set of tools. So I want to go over sort of a basic toolkit. And then I'm going to go throughout the video into more depth on these tools. So some basic tools we can use in our biotech toolkit are some restriction enzymes. You may remember from the last video, these basically cut DNA at certain positions, as you can sort of see right there. We also talked about ligase, which sort of glues different segments of DNA together. Plasmids and cloning is how we can actually clone or make multiple copies of DNA. DNA libraries or probes is where we're going to go to look for a segment of DNA. Then we've got some more advanced tools. PCR, which maybe you've heard in the news, this is a way of testing for a certain gene or making copies of a certain gene, making gene copies. DNA sequencing is telling us the order, right? It's A, C, C, G. We're figuring out the order of the nucleotides in the base. And then gel electrophoresis is a way, as you see here, of separating DNA that's been digested by restriction enzymes and visualizing it in a gel. Okay, so this was a brief overview, but what I'm going to do is I'm going to go through these tools in a little more depth so that you can learn some of our biotech tools. So I like to use the metaphor cut, paste, copy, find, like you would use in a word processor, like you're using Word or Google Docs. And these are some of the tools in my metaphor that I want to use. If we want to cut DNA, right, we actually use restriction enzymes. And restriction enzymes will cut at a specific place. If we want to paste two segments, this segment of DNA with this segment of DNA, we're going to use this enzyme here. It's called ligase, right? This one was a restriction enzyme. This enzyme here that glues things together is ligase. Copying is a little more complicated. If we want to copy a plasmid, right, a small circular piece of DNA, we can insert these into some bacteria. They can do the copying for us. Or we can use a fancy machine, a PCR machine, I'll go over more in depth in a moment, to make multiple copies. If we want to find a certain gene, we might use a probe to go and look on some place on the DNA with some little fluorescence so we can actually find the gene that we're looking for. Okay, so let's go over these one by one. Restriction enzymes I mentioned are basically ways of cutting DNA at certain palindromic sites. What's that mean? Well, if you look here, this restriction enzyme, here's an enzyme, here's some DNA. This enzyme will come in here and it will cut at these certain sites. So it will make us, like scissors, cut the DNA in half. So in here you can see it's cut there. Now we're left with two pieces of DNA, one there, because this restriction enzyme cuts the DNA. It leaves what we call some sticky ends, right? Because there's some nucleotides hanging over there. And those nucleotides will then be attracted to other nucleotides that match it. So it kind of makes it sticky. Now, where do we get these restriction enzymes? Did we just engineer them in a lab? Actually, no. We have found these in bacteria. So bacteria evolved restriction enzymes because what was happening was these bacteriophages, these nasty viruses, would land on bacteria and they would insert their viral DNA into the bacteria. Well, the bacteria didn't want it. So what they did is they evolved these restriction enzymes over time to cut up that foreign DNA with certain specific sequences, right? So a little more in depth, how these enzymes work. They cut the DNA at specific sequences. We call that a restriction site that the restriction enzymes go to. And it's odd, they actually cut in these symmetrical palindromic sequences. So let me give you some examples of these. If you've ever heard the phrase, Madam, I'm Adam, that's a palindrome. So palindrome, because forwards and backwards it's the same. Madam, I'm Adam. If you start at this end and go this way, Madam, I'm Adam. So that's palindromic sequences, and that is where these enzymes seem to cut. Now remember, I told you these evolved in different bacteria, so we actually name the enzymes based on the bacteria they came from. So obviously EcoR1, this restriction enzyme, came from E. coli. Now, how do these work? I sort of showed you this in the previous slide. The enzyme, it finds the palindromic sequence. So notice, we got GAATTC up here. If you look down here at the complementary strain, GAATTC going in this direction. Once it finds that particular site, the particular enzyme that's looking for that site will cut it up into different pieces of DNA, just like you see here. Here's another example. Right here, GAATTC backwards and forwards. So we can see that our scissors, our enzyme is going to cut in that specific location. Okay, so a little more complicated. So that's pretty easy. Now let's move on to some things that are a little more complicated. If we want to copy DNA, I told you we've got a few ways. One, we could insert it into bacteria, have the bacteria copy it for us. We do that to make several things like humulin, human insulin. But to understand how this works, we need to first understand about plasmids. Plasmids are small, self-replicating DNA molecules, and they're circular. So basically, to manufacture one of these, we take our DNA that we want, right? So if we want this particular piece of DNA to be manufactured, we would insert it into the plasmid and then insert this plasmid into some bacteria. So in other words, the plasmid is a vector or vehicle to get into the organism. We call this process transformation because what we're doing is by inserting this recombinant plasmid, and that's just a fancy word for it's a recombined some DNA, right? We take some DNA and put it here and they've recombined. into a bacteria so that the bacteria make lots of copies. And then we can grow that bacteria on an agar plate and then get lots of that gene. So this is what a typical plasmid might look like. So it's got sort of an origination point. Then we might want to insert a particular gene. And then we got to make sure there's a promoter to get this turned on. But if this is the gene we want, maybe the gene to make insulin, let's say, or a gene that we want our bacteria to glow in the dark. We do that a lot. We'll do a lab with that called green fluorescent protein. We'll insert a gene into a plasmid and then put that plasmid into the bacteria. Well, once this plasmid's in the bacteria, what do? Well, they make a bunch of copies, they replicate it, they transcribe it and translate it into proteins. This is how we're going to get the green fluorescent protein because we've inserted the gene to make that. So you see all the plasmids in here making lots of copies that we need. Because ultimately, here's what you need to remember. The DNA in the plasmid will code for the RNA, which codes for protein, and we know that protein is what gives us the trait. So in the example I just gave of glowing in the dark with this GFP, all we got to do is insert the gene into the bacteria. It transcribes and translates, and then we get the trait of glowing in the dark. Okay, so how do we know that the bacteria that we are trying to grow actually took the plasmid, right? Because I've been telling you we need to insert this plasmid into the bacteria. Well, how do we know the bacteria actually took the plasmid? Well, one way we do that is use a selecting agent. And we often use an antibiotic called ampicillin. Ampicillin is an antibiotic, which, if you think about an antibiotic, kills bacteria. Well, wait a minute. How will bacteria grow if we're putting antibiotic on a plate? Will the bacteria grow at all? No, they shouldn't. But we can actually change the bacteria to be resistant to the ampicillin. So how do we do that? So if you just take a normal plate that's got some broth on it, LB, we call it luria broth, you're going to get lots of bacteria growing. Look at all that bacteria growing. But if we then have a plate where we have the broth, but we also have ampicillin on the plate, our antibiotic, only bacteria that's been transformed will grow. In this case, the transformation, if you look, was both the GFP protein to make it glow, and then something we call a beta-lactamase gene, which makes it resistant to ampicillin. So the only bacteria that will grow are the ones that took up the gene. If it didn't take up the gene, like this one, let's say, notice it didn't grow on the plate. So we can be sure that the only ones growing were the ones that were transformed. Okay, so how do we make a bunch of copies of a gene? Let's say we're doing a crime scene lab, and we want to make a bunch of copies. We found a little piece of DNA at a crime scene, and we want to make a bunch of copies of that so we can use it in testing. Well, another way to do that is PCR. PCR stands for polymerase chain reaction, and it uses a thermocycler. Let me write this out. Polymerase chain reaction. So basically, it's a chain reaction using a polymerase to make a bunch of DNA. Well, you may remember what do polymerases do. DNA polymerase makes more DNA. So a PCR uses a machine called a thermocycler. Break down the word, thermocycler, right? It cycles through different temperatures. I've got one in my lab, but you can see a picture of it here. We also will use some primers, and then we got to put in some polymerase. So what are we doing? PCR is basically doing this. We've got some DNA that we want to make a bunch of copies of. There it is. It starts at a relatively low temperature. Then the thermocycler bumps up that temperature really hot to denature the DNA, to open it up. Once it's opened and denatured, then we insert these primers. Primers are basically like little probes that are going to go and find a certain area of the DNA we want. So basically, we're making a complementary copy to match the gene we want. Then the temperature raises again, and Taq polymerase, this polymerase right here, actually then starts to extend the primer and make more copies of the DNA. Okay? So this is how PCR works. Basically, changing a bunch of temperatures, which opens up the DNA, lets the primers join, and then extends that DNA into many, many copies. Then what we'll do is it will start this whole process over again, I don't know, 50 to 100 times, so that it makes a bunch of copies of that DNA. Okay, so how do we locate the gene of interest? I sort of just alluded to that. We can use primers. We can also use a process called DNA hybridization. If you break down the word, DNA hybridization, it sort of sounds like we're making a DNA hybrid. So it's sort of half DNA and maybe half something else. And this is where we might use something called a probe. A probe is often something that is a short single strand of DNA, right? You can see the probe right here. Short single strand of DNA. And it is complementary to a gene of interest. So let's say we want to find the insulin gene, right? So here's my gene for insulin. So this probe will have a sequence of DNA that's complementary, so it will come and match. Now, what do we do to that specific probe? We also tag it with some radioactive phosphorus, right? We know DNA has sugar and phosphate in the backbone. So this will make it fluoresce, and we'll be able to recognize it once it's found. Okay? So the hybridization occurs when the probe and the DNA join together. We can find it because it's sort of glowing. It is fluorescing with that radioactive phosphorus. Okay, so let's do something a little advanced now. Let's say that we needed to solve a crime and we want to compare DNA. So let's say we found some DNA at a crime scene, and let's say we found a suspect 1 DNA and a suspect 2 DNA. So we've got some crime scene DNA, and then we've got our DNA from 2 suspects that we have, and we want to see if the suspect's DNA matches the crime scene DNA. Well, what we're going to do is a process called gel electrophoresis. This is going to separate the DNA based on size. And we also use these little things called restriction fragment length polymorphisms. Not super essential that you know what that means, but just know this is what we're going to create by digesting these DNA. So what we do, we take some restriction enzymes, remember those cut up DNA, and we take those restriction enzymes and we apply it to all the DNA. So basically the crime scene, the suspect's DNA, it gets digested into these small little fragments, which is what you see right there. Then we take those fragments, after they've been cut up with the enzyme, and we put them in these little gels. Well, we're checking to see if there's a polymorphism. We're checking to see if there's some similarity. So we take this DNA, they've been digested with the enzymes, and we put them in all these little wells, okay? These little holes in the top of the gel. Then we take that gel and we put it in an electrophoresis apparatus. That apparatus runs current, electricity, from this direction to this direction. Well, that DNA often is positively charged, and as what we see here, when we run our electricity, it's going to take a lot of that DNA and move it down. So what we're going to see is that the DNA will move through the gel. Now, if you think about this, if you're a large piece of DNA versus a small piece of DNA, which one do you think is going to be able to move farther? Well, we actually know it's the smaller pieces that end up going farthest, right? So when you're looking at a gel electrophoresis, the smaller chunks or fragments of DNA are down here, whereas the larger fragments of DNA are at the top. All right, so what's the point of this? Well, then the point is to compare. You look at this lane versus this lane versus this and this and this, and see if you find any that are the same. So when I'm looking here at these compared to each other, I'm not seeing any that are identical. This one has a thing here, and there's one there, so maybe we got some similarity there. These two have a line there. These have these shared. So this would help us to determine which of the DNA lines matched the crime scene line, and that would allow us to solve the crime. All right. So let's wrap this up by taking it back to the very beginning. I talked to you about GMOs. This method that we've just talked about, all these tools, allow us basically to take little bits of DNA from one organism and put them to the other to genetically modify an organism. Why would we do that? Well, here's a few reasons why. One, we can actually insert genes to make certain plants give us more nutrients. Prime example of that is golden rice, which was a genetically modified rice, which engineered to provide more vitamin A through the process of giving more beta carotene. So we could actually insert genes for nutrition into different plants. We can also make the plant grow better, right? Make it more hardier, resistant to certain temperatures, resistant to certain diseases, okay? Also, because we're getting more yield, because we're getting plants that survive longer, it's actually quite cheaper to use genetically modified organisms. And we can also take and introduce genes that are pest resistant. So we can do a gene that gives a certain pest resistance to a certain crop. So therefore, if some caterpillar or some bug came to eat that crop, it would get sick, right? So they're pest resistant. Okay, so lots of pros to GMOs. Now, I feel I do got to bring up a few cons just to be fair. Some of the people arguing against GMOs would say, well, we don't know what genes you're inserting. We might have an allergic reaction to one of those genes now you put in corn, right? I could eat corn before, but now you've put in some gene from a peanut or something and I'm allergic. All right, so that's a real concern. However, most of the companies that manufacture these GMOs heavily test these crops. In fact, GMO crops are tested by far more than any other crop just because they're done by a company, right? And they don't want to get sued. We also, another con people throw in is this idea of superbugs. Well, if we keep making our corn resistant to bugs, the bugs will evolve and they'll be resistant to the new corn that we've made or the new rice. Okay, right? Also a legitimate concern. However, if you think about it, the same thing would go to our natural pesticides or our chemical pesticides, right? So if we keep spraying pesticides all over these crops, well, the bugs either way will become resistant to those pesticides. So both of these really are, in my opinion, outweighed by the pros of GMOs. In fact, you may know that 92% of corn and 94% of soybeans grown in the USA actually today come from genetically modified seeds. So obviously you get to make your own decision about what you put in your body and what you eat. But if you're looking at the scientists, most all scientists agree that GMOs are perfectly healthy, perfectly fine, and if anything, actually do more for the world. Okay, so what's the deal with GMOs? There you go. You got a few answers to think about. How do we get to this point with GMOs? Because of science, because of all of these biotechnology tools that have allowed us to really grow some things and to change and manipulate organisms that make them better for us.